So today we will talk about in our lecture about the glomerular filtration barrier. So the very first thing to understand the glomerular filtration barrier structure and then the function is to understand some context for various things which either participate to make a structural barrier or they participate in its function. So the important things to understand in our context are: number one, the characteristics of the albumin; number two, characteristics of glycosaminoglycans, what are these things and how do they operate in our context for the glomerular filtration barrier. Then what are proteoglycans, what are glycoproteins, what is glycocalyx, what are sialoglycoproteins. So these are the various things which we should know before we can understand the glomerular filtration barrier. So having said that, let us talk about the albumin first of all. So albumin is the most abundant protein in the plasma. The most abundant protein in the blood is collagen, but the most abundant protein in plasma is albumin. The size of albumin is about 3.6 nanometer. This is very important to understand the glomerular filtration barrier. So please do not forget this size is about 3.6 nanometer. The other thing which is really important for our context, it is a negatively charged protein. Most of the plasma proteins are negatively charged. So albumin is not different. That also is negatively charged. So these two things are very important: the mass, the plasma half-life, and characteristics. I just put them over here to give some characteristics of the albumin, but important thing is size 3.6 nanometer and charge negative. So having said that, let's talk about the glycosaminoglycans and what these are. In a summarized way, I can say that glycosaminoglycans are the glucose or sugars which are connected with each other to make long chains. These long chains are usually negatively charged. So when these negatively charged chains are attached to cells, then those cell surfaces or wherever these things are attached, they become negative as well. So to understand glycosaminoglycans and proteoglycans and glycoproteins and glycocalyx and sialoglycoprotein, what we should understand is we should start from the basic building blocks, and the very first building blocks which we should know are the carboxyl group. So carboxyl group is basically a combination of carbonyl group, which is a carbon which is attached to the oxygen, and then hydroxyl group, which is OH. And hydroxyl group, as you know, is a negatively charged group as well when it is found in the ionic form. So here if you see, this is a carboxyl group in which a carbonyl group is attached to a hydroxyl group. Of course, there is going to be a side chain attached here as well. That is less important for our discussion at this moment. Then, of course, glycosaminoglycan, because they contain this hydroxyl group, they are negatively charged at physiological pH. The organic molecules which have gotten hydroxyl groups are normally called alcohols. The other thing is this, that whenever we have charge on any molecule, that molecule will be loving water. Water, being a polar molecule itself, would like to be around the negatively or positively charged ions. So here in the in the carboxyl group, the hydroxyl group itself is negatively charged, which then loves water. The other thing which we should know is acetyl group. So acetyl group is a methyl CH3 which is connected to a carbonyl group, carbon attached to oxygen. So here is a acetyl group present in front of us. Here for this question mark, you could have various things which are attached here as a side chain. In our context, these are going to be the glucose or sugars. The sugars which have gotten amines are normally called amino sugars, and usually these sugars are acetylated, which is the molecular structure here attached to them, and that removes the positive charge from these amino sugars. So if we see here for a second, this is a glucose which is an amine. So this is a glucose molecule which has an amine attached to it. And normally this amino group which is attached to the glucose imparts a positive charge. If we acetylate this, so if we attach an acetyl group here, that would remove the positivity from this amino group. So that is how this glucose or this amino glucose will become not positively charged. Now coming here, some characteristics of the glycosaminoglycans. So they are negatively charged, as we just saw that they contain the hydroxyl ions which are negatively charged. Then their amino group are acetylated, so their positivity is reduced or removed. And then they also have, as we would see a little later, they have sulfate groups attached to them which imparts strong negative charges as well. So the characteristics of the glycosaminoglycans owing to these facts is that they are negatively charged. The other thing are they are slippery. So it's a very interesting phenomena. You might have noticed that when you bring the similar poles of magnets together, for example south end with the south end, you see that they normally they repel each other. Not normally, always they repel each other, and it feels like they do not want to come near. Similarly here, the negatively charged branches or molecules, when they are brought near each other, they slip past each other and they repel each other, and that gives us a slippery feeling. So glycosaminoglycans are normally slippery. This slippery or negatively repelling function or repelling function due to negative charge is very important when it comes to the foot processes of the glomerular filtration barrier, because these foot processes are covered by the glycosaminoglycans, and then they repel each other, and so they create the slit pores or little gaps. So we'll see that this is a very important factor. Then glycosaminoglycans are also squishy. That is because they are negatively charged, so they are hydrophilic, and so they attract a lot of water with them. So we will continue in our next lecture about the structure of the glycosaminoglycans, glycoproteins, and proteoglycans.